So in this video we're going to chat about mitosis, and in this rather mesmerizing video we have the image of a frog's egg that has just been fertilized and it is undergoing mitosis, and it is dividing over and over again because one of the important roles of mitosis is to allow growth in multicellular organisms. So one cell becomes 2, becomes 4, becomes 8, becomes 16, and eventually becomes the billions of cells needed to make a multicellular organism. On top of that, mitosis is important for repairing or replacing worn-out cells. So, for example, you need new skin cells all the time, you need to replace them. And another thing we can use it for is asexual reproduction. So if you want to make more of a cell, because maybe you're a single-celled organism, maybe you're an amoeba swimming around in a pond and it's a really favorable environment. You want to divide, make more of yourself and take advantage of that successful environment. So in mitosis we make identical cells, so genetically identical cells, and each time one cell divides and becomes 2 identical cells. So let's have a look at some cells under the microscope. So these are some images from an onion taken at the root tip. The root tip of an onion is a very quickly dividing area, and you'll notice that all the nuclei look a bit different. So we can actually see visibly here some chromosomes. So you can see here some chromosomes, and here we've got some chromosomes, and there seem to be some odd things going on. This is what we'd expect to see a normal nucleus looking like, a very clearly defined cell nuclear membrane, and then most of the inside of the nucleus isn't particularly interesting, but there are some darkly staining bits. But over here, this does not look the same. So let's have a closer look, a more cartoon look at what's going on during mitosis. So in this image here, we have the 4 phases of nuclear division in mitosis. So we have prophase, metaphase, anaphase, and telophase. Now in prophase, that's the first stage. What's happening is you can see the nuclear membrane is breaking up. So we've got a little kind of remnant of the nuclear membrane, and we can see the chromosomes clearly visible. Now, chromosomes are made up of 2 sort of arms, and they're held together by a centromere. Now, the arms are called chromatids. So there are 2 chromatids, and the central piece is called a centromere. And you can see that there it gives us a characteristic X shape that we think of when we think of chromosomes. And in this particular cell, we've got 2 pairs of chromosomes. So we've got 4 chromosomes altogether. So in prophase, the chromosomes become organized and become shorter and fatter, visible as this X structure, clearly defined centromere, and you can see the nuclear membrane has disappeared. Also, at the top and the bottom of this cell, we've got the centrioles, and they are organizing a structure between them called spindle fibers. And these are going to be really important in the next phase. So you can see in metaphase, the chromosomes have arranged themselves on the equator of the cell, and the centromeres are actually attached to spindle fibers, both up and down. And you can see in the next phase, it's a very dynamic phase, the chromosomes have been ripped apart at the centromere. So one of these legs has gone north and one has gone south, and they are being dragged north and south to the poles of the cell by their centromeres attached to the spindle fibers. The last stage, telophase, is where we start to look like a cell again. So we reverse some of those prophase stages. The nuclear membrane starts to reappear, and the chromosomes start to decondense. They are no longer short and fat. They are becoming unraveled because they're going to be just part of a normal cell doing their normal work. So you have to be able to access the DNA. So those are our 4 stages PMAT, PMAT for short. So let's look again at a picture underneath a microscope, and now you can see some of those stages of cell division of mitosis. So over here we've got the chromosomes being pulled apart, that's anaphase. Here they're lining up on the equator. Here they're becoming distinct, so that's prophase. And here is interphase. This is the bit where it's not actually dividing, that's when it's just being an ordinary cell. Let's have a look. I can't really see telophase too clearly on this one, but if we go back to the big picture here, I think telophase has just happened here because you can see how close together those two cells are there.